Namaste students, हमेशा खुश रहो बेटियों. I am JV'n's Dr. Neeraj Dholia and we are here to study the developmental biology subject. The topic which we'll discuss today is the branches of the embryology. So in the earlier sessions, we have understood that what is the developmental biology, and we have also understood that what is the uh, history of the developmental biology. So in the previous session we have seen that the developmental biology is divided into two parts, that is prenatal and postnatal development. We have also studied that the history of the embryology, how the embryology is developed. Now next we will see that what are the different types of the embryology. Okay, so we will start from the beginning, that there are 5 types of embryology. So the 5 types are the descriptive embryology, comparative embryology, experimental embryology, chemical embryology, and teratology. So these are the various types of the embryology. So let's understand one by one what is that. First of all, we will see the descriptive embryology. So as its name suggests, description, it gives the description of the embryology. So this field of embryology is associated with the morphological description of different embryonic stages in the ontogenic development of individuals of different species. So morphological description of embryology, that how the morphology of different embryos varies, how a specific type of organ will evolve, and how it appears in the initial stages, and what are the changes will occur in specific type of regions, and how it is uh, complete the process of the development, and how it is the finally it is developed into the in a complete animal. So it describes each and every step of the embryological development from the zygote to the complete animal before birth. Means ontogenic development means, ontogenic is the prenatal development. Prenatal development means the development before birth. So before birth, what are the changes occurred are explained in the descriptive embryology. So the descriptive embryology is a very important for the study or study. We can compare the uh, phenomenons once after the second. So we can if we know about the stepwise or description of the embryology, so we can control them. Okay, if you know anything, then you can control that. If you are not knowing any pathway, you cannot control. So if your pathway is known, we can regulate it by many chemicals, inhibitors, and drugs. Okay, so descriptive embryology is important field of the or branch of the embryology. So next type of the embryology is the comparative embryology. So comparative, as its name suggests, that it compares. It embraces the comparative study of embryology of different animal groups. So why it is important? So it is important because the comparison give an idea about the development or the evolution. So evolution from an eukaryotes, from a prokaryotes to an eukaryote or up to a mammal. How it evolved? So the comparative embryology describes the changes and what the changes occur from one animal group to the next level of the animal groups. There are various types of the animals, from the invertebrates to vertebrates to the higher vertebrates. Okay, so embryos are there. So in embryological development, what are the changes? Like fishes, birds, mammals, okay, in amphibians, the embryos are there. But at the prenatal development or in the ontogenic development, how the embryo develops from the one cell to the complete animal. So it is studied under the comparative embryology, and it will answer us many things linked with the evolution. Okay, so it is the comparative embryology. Next one is the experimental embryology. As name suggests, that it involves the experimental studies. Okay, so it involves all those studies that attempt to understand the various fundamental mechanisms in the development of different animals, like fertilization, cleavage, gastrulation, embryonic induction, determination, and differentiation. So these are the various phenomena which occur during the embryological development, and uh, we can conduct the experiments at each and every step, and which will be explained by the experimental embryology, and they all will come under the experimental embryology. So if for example we in the differentiation process, what is the differentiation is the um, maturation of a undifferentiated cell to a dedicated cells. Like the stem cells are the undifferentiated cells, and when they are converted into the skin cells, that is we call it is a differentiated cell. Okay, so skin cell do not have the potential to multiply or renewation as the stem cells have. So these the differentiation experimentally we can isolate the stem cells, and we can conduct few experiments that control the differentiation, and you can study the any drug or any chemical which affect the differentiation steps. Okay, likewise the fertilization, cleavage, gastrulation, embryonic induction, and determination, all these steps can be explained using the controlled condition in the lab and explained by the experimental embryology. So all the experiment will come under the this category. So next one is the chemical embryology. Okay, so it involves the branch of it is a branch of embryology, includes all those studies which employ various biochemical, biophysical, and physiological techniques for understanding the embryological events. So to understand the embryological events, we use various things. Okay, we can use the biochemical things, we can use the biophysical instruments, we use the physiological techniques to understand the embryology. So these, when you use these things to understand the mechanism at molecular level, so we will say is chemical embryology. Okay, तो chemical embryology के जो है, this is used to understand the embryological phenomena at the molecular level. Okay, so molecules means chemicals. Chemical embryology, if we come to the words, chemical embryology means we use certain chemicals. Okay, and molecular level, molecules are also the chemicals. So next is the teratology. So teratology is generally defined as the embryological defects. So it is a branch of embryology concerned with the study of malformations of birth defects. Okay, malformations means the underformation or the overformation of the disappearance of few organs or fusion of some organs at the time of the embryological development, collectively known as the birth defects. So the substances that cause birth defects are called the teratogens, and that's why this is known as the teratology. So study of teratogens on the embryological development is known as the teratology. So the it is very important because there is a significant percentage of the birth defects found in the India as well as at the global level. So these are the different types of the branches of the embryology. So this session was powered by digital version 2 Jyoti Vidhyapeeth University. Hope you are satisfied with my digital session. If you have any query, please write on the comment box. I will definitely resolve the problems. Thank you.